What happens? Well, what happens if you do have just a true incompatibility problem? You can't figure out the cDNA is not working, reverse transcriptase isn't working, you don't know what to do. All right, so instead, we figured out how to use cloning vectors of eukaryotics. Okay, so the first one that we used was yeast. All right, Saccharomyces cerevisiae is what it's talking about. They're called yeast artificial chromosomes, or YACs. Okay, they're vectors with an origin of replication. They have a centromere. They have 2 telomeres with foreign DNA. Okay, and then so it's got everything that you need so that it will behave exactly the way it needs to in mitosis. All right, so mitosis makes identical cells. It will do exactly the way it's supposed to in eukaryotes with mitosis. And so the reason why they came up with yeast is because they're as easy as bacteria to grow, right? Because they're single-cell organisms. And they're one of the only eukaryotes that actually has plasmids in it. Okay, so we use those as the actual cloning vectors. Okay, now we've got that transcription thing taken care of. What about translation? Because remember that eukaryotes are they're just divas. They're basically divas. They have to have all this modification stuff. Remember post-translational modifications where you might take out some codons and some other stuff? All right, eukaryotic proteins are going to need to be modified after they've been translated, right? So bacteria and yeast don't do this, right? They're single-celled organisms and they don't need to do that stuff. So now we have to use a plant or animal host cell to do this. All right, so, but here's the thing. We could use a vector that is plant or animal, all right, but how are we supposed to get the plant or animal to take up foreign DNA, all right? Because usually plants and animals will not do that. They really don't. They're not very efficient at it. They don't need to because they usually they have everything that they need right there in their cell. Okay, so how do we force it to take up DNA? All right, it's something called electroporation. It's basically a shock, all right? It's electrical shocks. And what you do is you put a brief electrical pulse on the cell that allows the plasma membrane to create holes in it temporarily, and so that the DNA can, be sucked in or slide in or enter into there. That's one way, electroporation. The other way is to directly just inject it, okay, with microscopic needles, which is what I used to do with the cells in my worms that I used to work in the lab, which I will be showing you when we take our field trip there. Okay, so you can directly inject the DNA into the cell by like a little microscopic needle, or you can shoot it into the cell, okay, which we call this bombardment. All right, so basically you have DNA that is going to be attached to these microscopic beads. All right, they're usually made out of metal, like gold or something like that. Gold usually is what they make them out of. And so you attach these you attach their DNA to these microscopic particles, and you put them in this gun. It's not like a gun. It's like, it looks like a Tommy gun, except it shoots out of like 4 or 5 barrels. Okay, so one of those. And basically what it does is the beads will enter into the cell, and it will carry with it the DNA. Okay, so we did this with the worms. I've done it with worms before. It works successfully. They usually do this with plants because plants are very sensitive to electroporation, and it's really hard to get past that cell wall to DNA inject them. So what they do is they just shoot the crap out of them with DNA. Okay, now recall back that you have isolated the recombinant DNA that you need, right? But you still have these well, not millions, but thousands of other different types of recombinant DNA. You may need them in future, okay? You don't know because you're looking at this gene only, and maybe this gene leads to this gene, Oh, hey, well, you've already got the recombinant DNA already there. It's already been made in the plasmid. You don't have to do it again. So what do? You store it, right? It's called a genomic library. And basically, it's called the shotgun approach, okay, to making recombinant DNA, where you just cut it all up and you mix it all together and you make 1,000 different recombinant DNAs, and then you try to figure out which one is the one you want to study right then. Well, what do you do with the rest of them? Well, you save them, and that way they're there when you need them the next time, okay, if you need them ever again. And they call it a genomic library because it is a collection of all your recombinant DNAs that you've already made so that you don't have to go back and remake them again. Okay, so it's many, many fragments that have been made into recombinant DNA. You're not sure which one you may need in future, but it's there if you do. Okay. Okay. So another thing you can use plasmids. Sorry, my dog is. Come here, Evie. Come here. Come here. She's distracting me. Here's my puppy. Isn't she cute? Stinky. My little baby. Okay, enough. Alright, sorry. So, alright, so you have these recombinant DNAs that basically make up your library, okay? And you can use plasmids as the cloning vectors. You can use bacteriophages as the cloning vectors. You can use YACs as the cloning vectors. You can also have cDNA libraries. Okay, I don't know if you've ever heard of those, but they're very, very common. They're limited because it's the DNA that has been complementarily made by mRNAs, and that doesn't really happen that often. But so they're limited because the only genes, the recombinant DNA that you have are the ones that have been transcribed already. Okay, so you do have cDNA libraries; those are available, and they're more limited. They're more limited than the regular genomic libraries. Okay, so we are moving into page. Let's see here. I've just been rattling off 382. So we've only gotten through like 5 pages maybe, and I've been talking for 32 minutes, so I'm sorry. I'm going to stop rambling. I'm going to try to get through what I need to get through to talk to you about, but all right, let's go. All right, so say you don't have time to wait on this one piece of DNA through these plasmids, or say your DNA source is impure, all right, or you have minute microscopic amounts of it that you cannot afford to lose. All right, say it's a murder trial and it's a rape murder trial, okay? You've got a limited amount of semen and you've got a limited amount of time, right, before this guy gets off, okay? So you have to be able to run this in a gel, you have to be able to analyze all this DNA, but you don't have enough. You have like 1/1000th of what you're supposed to need, okay, to be able to run any kind of analysis on it. And this guy's about to make bail, and he's probably going to skip town. Okay, so what do? You can't wait around for bacteria to start cloning it, and because that could take days and days and days. That can take about 3 or 4 days. So instead, there is this method that they came up with in 1985 called polymerase chain reaction, or PCR. All right, it's a way to rapidly replicate small or impure amounts of DNA by a 3-step cycle. Okay, so you need a specialized DNA polymerase, which we're going to talk about why it's specialized in a second. And you need a supply of nucleotides to build your new DNA strands. And you need primers, okay, so that the polymerase knows where to hook on and start replicating. So this would create exponential growth because it's like regular DNA replication. 1 strand creates 2 strands, 2 strands creates 4, 4 creates 8, 8 creates 16, and so it's exponential growth. So by the time you have in a 24-hour period, you could have billions of pieces of the same DNA that you've been needing. And then you can do your analysis on it in a day, and he goes to jail, okay, for the rest of his life, which would be great. So one of the pros of this is that it's speedy, okay, so you can do this in about 12 to 24 hours. You can replicate it a billionfold. That is ridiculous, okay, that is a lot. That is very, very efficient. Also, since you have primers for that piece of DNA, because usually you know some of the sequence of your gene, you know what the sequence of your gene is, you can create primers. And so then that means that the DNA polymerase is only replicating your piece of DNA that you need. Okay, and so, and then the other thing is that the starting material, it can be impure, it can be partially degraded. I mean, it can be crap, and this can work. Okay, so this is a miracle worker. All right, so The cons about it, they can't really produce a whole. Okay, a billion-fold is a lot of DNA. Starting from 1 to a billion is a lot, but it's not a lot to do any sort of like long-term research on. Okay, in that case, if you're trying to do that, you would rather it be done in a bacterial chromosome or in a bacterial plasmid, because this method can make a lot of mistakes. All right, it's DNA polymerase. And remember that it is very efficient, but the more times it has to replicate, the more times it has to mess up. And so there are mistakes that can be made, and that can kind of sully your genes, all right, because some of them will be wrong. And also, it can, for the criminal, for the sake of my little rape and murder story, if some of the DNA is not correct, is not completely identical, he could walk. Right, because it's not his, right? Because there's 1 or 2 base pairs wrong, different. It's not his. So you have to be careful. I mean, it's good if you need like a quick fix and you just need to run some analysis on it, but it's not something that is really going to be useful if you need a lot of DNA. Okay, so we figured out how to replicate our DNA. We figured out how to get it in large amounts. We figured out how to get it very fast. We figured out how we can clone and express it in different vectors. Now, what do we do with it? Well, we got to analyze it now. So this is where genomics pops in. Okay, genomics is a study of all the genome of a whole organism and all the interactions of that genome to make the organism the way it is. Okay, before you can do any kind of specific analysis like sequencing or something, there's always more general comparisons that you can make, like say for size or charge, and this is by gel electrophoresis. Okay, so if you read about it, it's figure 20.8, page 384, and basically what you're doing is you're looking at a piece of DNA based on its size or based on its charge, and you can also do this for proteins. Okay, so you would restriction enzyme that sucker. Okay, and then you would run it in a gel, and the slower or the bigger proteins are going to run less. They're going to run less fast, so they're going to be at the top of your gel. And then the smaller ones that can run faster are going to be at the bottom. Okay, you shock it with an electrical current. The positive ones are going to move towards the negative side. The negative ones are going to stay away from the negative side. Okay, so you can separate it by size and you can separate it by charge, right? And this holds true for proteins as well. Okay, size and charge. So. Now, let's see. You've got, all right, on figure 20.9, so when you run it in this gel, okay, the first thing that you're going to do is you're going to restriction enzyme it, right, which isn't really a verb, so I should probably say you're going to use a restriction enzyme on it, okay, and it's going to cut it. And remember that restriction enzymes are specific, they're site-specific, which means it'll always cut it in a specific pattern, right? You'll always get this many fragments. And then you run it in the gel, and you should be able to see what the banding pattern looks like. Okay, so this is a good way to compare an unknown piece of DNA with your DNA. Okay, if you want to know, is my DNA the same? Is this my DNA? Then you would run it. If the banding pattern is the same, then it has more of a likely chance that it's going to be yours. Okay. The patterns are going to be characteristic of certain restriction enzymes. Okay, so that means that they are always cut at the specific site. If you have this DNA piece, every time that you cut it, it's going to be specific for that restriction enzyme. You run it on the electrophoresis gel to see them, and then you can recover them out of the gel. Okay, so you would actually physically cut them out of the gel, and then you can basically isolate it again. Okay, you purify it back. It can tell you subtle differences in nucleic acid sequences. Why? Because if you have a bigger sequence, it's going to be a bigger piece of DNA, which means it's going to run at the top. Okay, so if I have a banding pattern here, and I have a banding pattern here, and this is the top of my gel right here, I know that this one has more nucleic acids in it, okay, than this one. Okay, I need a break. And this would be about where the bell would ring for lunch anyway. So I'm going to let you guys, I need a break real quick, and then we'll come back. And when we come back, we'll start talking about southern blotting. All right?